All right, so let's move on to actually talk about what these biomolecules are. So these are also called macromolecules, again, molecules that must be synthesized by living things. They're primarily made of carbon, but we've got other atoms involved in different proportions and different ones. We have oxygen, hydrogen, nitrogen, and then we can also have things like sulfur and phosphorus as well in smaller quantities. The reason that carbon is the central atom here is that carbon is a very rigid, very fixed molecule. It allows for 4 covalent bonds, so that's going to be a good amount of structural stability, structural rigidity offered to these molecules. So all of them we will see have mostly carbon. And so this gives us 4 main types. Again, when we mix these atoms in different quantities and different proportions, we get 4 types of macromolecules or biomolecules: carbohydrates, lipids, proteins, and nucleic acids. So what we will do today is speak about each of these categories. We'll talk about some of the structural properties and how those structural properties lend themselves to some functional differences. And what we will explore is that, for example, lipids are very different functionally than carbohydrates, for example. We know that lipids like oils and fats don't play nicely with water. So we're going to look at the molecular reason for that and then talk about some of the other differences and functional properties of each of these as well. So starting off with carbohydrates. So carbohydrates, we typically associate these with things like, our diet, bread, pasta, rice. These are macromolecules that are composed of carbon, hydrogen, and oxygen. There are 3 main types. We've got monosaccharides, disaccharides, and polysaccharides. And again, this is just the different type based upon the number of these monomers that are attached. So monosaccharides are the single monomers or single units of carbohydrates. The examples here are glucose, fructose, which is fruit sugar, or galactose. If we start to combine these monosaccharides together covalently, we then form disaccharides, which are slightly longer saccharides. If we combine glucose and fructose, for example, this gives us sucrose, which is our table sugar. If we combine galactose and glucose, this gives us lactose, which is milk sugar. And so disaccharides are slightly more complex than our monosaccharide units. We also have polysaccharides. So these are even more complex. Things like glycogen is a polysaccharide. And as we can see here, glycogen is nothing but many glucose monomers linked together over and over. Glycogen is also one of our storage molecules or storage reserves for energy. This is how we store glucose away. We store glucose in our liver, in our muscle, and this is what it basically is, just branches of many, many units of glucose that are covalently bound together. Okay, let's talk about the process of how these bonds are made. So in order to synthesize a saccharide, we have to covalently bind 2 monosaccharides, or 2 of those monomers. This is a process called condensation. So we add 2 monomers. An example here is glucose and fructose together, and then we release water as a byproduct, and we covalently bind these 2 monomers together, and that gives us a slightly more complex saccharide. In this case, that's going to be sucrose. If we want to split or hydrolyze this, this macromolecule, this more complex saccharide, we simply insert water and we break that covalent bond, and then we end up with glucose and fructose on the other end after having inserted water. So this is the difference between a condensation reaction that joins 2 monosaccharides together and releases water, and a hydrolysis reaction that splits these saccharides into smaller monomers and actually consumes water. Okay. All right, so that's the extent of what we will speak about for lipids. I will point out that they're one of the most important macromolecules. They're the most abundant. They also make up the largest energy source. We know glucose is one of the main energy sources for our cells. And so we're going to come back to carbohydrates again when we speak about glucose and the process of cellular respiration, and talk about how we actually take a molecule of glucose, use it to liberate energy, to liberate ATP so that we can do cellular work, cellular processes, build bones, build muscles, all those cellular processes that must be done, need energy, and we liberate that energy mainly from glucose, which is a carbohydrate. Let's move on to lipids. So lipids are, as I mentioned earlier, very different functionally from the other three macromolecules. They're composed primarily of hydrogen and carbon, not a lot of oxygen. And because they don't have a lot of oxygen, this gives them a property that we call nonpolar or hydrophobic. So if you think back to one of your earlier chemistry classes, maybe bio of the cell or so, you would have known that oxygen is referred to as an electron hog. Oxygen likes to grab electrons, and so molecules that have a lot of oxygen in them usually have an unequal charge around the molecule, and we call this polarity or being polar. So part of the molecule has a slightly positive charge, part of the molecule has a slightly negative charge because of this greedy or this hogging nature of oxygen that likes to grab or hog onto electrons. So because we don't have a lot of oxygen here in lipids, we see that there's a nice, even, stable charge around these molecules. We call them charge neutral, and we also can refer to them as being nonpolar. And because they're nonpolar, they also do not interact nicely with water, right? Something that I'm sure we know in our everyday lives. If you put some oil in water, they immediately separate. And that is because of this very small molecular difference, the absence of oxygen here, which gives lipids a nonpolar characteristic, or what we call a hydrophobic characteristic. Now, the 5 classes of lipids are triglycerides, ketones, phospholipids, eicosanoids, and steroids. So we're going to talk about these in a bit more detail going forward. So the first class, triglycerides, is really made of a glycerol backbone, which is a 3-carbon alcohol. And in this little drawing here on the right, we can see that this is that backbone, or that 3-carbon alcohol. And then attached to that are 3 fatty acid tails, or 3 fatty acid chains. These are simply long carbon acid chains, just carbons bound to carbons, bound to carbons. And so this is the structure of a triglyceride. And as you can see, because it has those 3 tails, is where we get that, that tri in the word triglyceride. Now, among fatty acids, we can have slight differences here structurally that bring about large differences functionally. So we can have a difference between a saturated fatty acid and an unsaturated fatty acid. So at the top here, this is a saturated fatty acid. So none of the carbons are bound by a double bond. So if there are no double bonds and there's no opportunity to bind more hydrogens, then we call this saturated. It has all the binding that can be done, or all the covalent bonds that can be formed. Whereas we can compare that to an unsaturated fatty acid, which are examples here at the bottom, that do have double bonds somewhere in between. Okay, and so they can have additional binding, additional covalent bonds. Now, if we go a step further and look at unsaturated fatty acids, we have another slight structural difference that creates a very large functional difference once again. So depending on what face of this fatty acid we have a hydrogen bound, that will determine whether it's a trans fatty acid or a cis fatty acid. So if we've got the hydrogens bound on the same side, then it's a cis fatty acid. Whereas if the hydrogens are on opposing sides, then it becomes a trans fatty acid. So this very small molecular detail is what creates the difference between a healthy fat versus an unhealthy fat, right? Typically, we associate saturated fatty acids as being slightly more unhealthy than unsaturated fatty acids. And then even among unsaturated fatty acids, we consider our trans fats. I'm sure you've heard that in everyday, trans fatty acids are no good for you. These are the fatty acids that we associate with plaque formation, which eventually leads to clot formation in vessels and heart disease. So the small molecular detail of having a hydrogen on the opposite side of that double bond is what creates this large functional difference and makes one molecule slightly more unhealthy and the other molecule not as bad for our health. Okay, so this is kind of what we want to point out, is that the structure is really important to function. All right, the third class of lipids, or I'm sorry, the second class of lipids here are ketones. Now, ketones are basically byproducts of fat metabolism. They become particularly important for diabetics or for persons on low carbohydrate diets. If you think about, and I think this is kind of big in the media now, ketogenic diets, or diets very low in carbohydrates, also tend to generate lots of ketones. So when we hydrolyze triglycerides, which are found in fatty tissue or adipose tissue, we then liberate fatty acids in the blood so that we can use those to make energy. But as a byproduct, some of those fatty acids are converted to ketones by the liver. So this can be, again, a desired effect. Certain conditions require very low carbohydrate diets or more of a ketogenic diet. And then diabetics have no choice. They also fall into this category of building up quite a bit of ketones. And if you think about it, just to kind of go into the clinical bit here a little, the definition of diabetes is if we have excess glucose in the blood. So we can have two types of diabetes, mainly type 1, where there's a problem with the pancreas and we don't secrete enough insulin. We tend to see a lot of ketones building up in the blood, and this leads to a state called ketoacidosis. Okay, this can be life-threatening, this can be detrimental, because of the fact that we want to keep our blood pH at a very stable level. If you think back to our discussion with homeostasis. And so if we have a lot of these ketones circulating in the blood, because they are acids, they will then shift the pH of our blood, and this can bring about a whole bunch of detrimental, even fatal consequences. One of the side effects that we see in ketoacidosis is acetone breath. So these patients will sometimes complain of having a metallic taste in their mouth, or you may even smell that metallic taste in their breath as well, or metallic scent, excuse me, in their breath. When we look at the structure of a ketone, it's simply a carbon double bound to an oxygen, and then we got two functional groups attached here. These can be X different groups. We just have an R representing that these can be two different groups attached. So one example of a ketone is acetoacetic acid. And this is specifically a hydroxyl group on one end and then a methyl group on the other. But we can have a lot of different types of ketones that have a lot of different variety in these functional groups, and will obviously offer slightly different functional properties because of these small molecular changes. All right, so that is ketones. So the next time you hear about a ketogenic diet or ketosis, I'm sure you'll understand exactly what that's referring to. The third class of lipids here are phospholipids. Now, phospholipids are very, very important. They have a property that makes them amphipathic. And amphipathic means that a part of this molecule likes water. We refer to it as being polar or hydrophilic, philic meaning that it likes water, and a part of the molecule, the tail of it, is the opposite. It's hydrophobic, it doesn't like water, or we can also refer to it as being non-polar. So we have this very interesting dynamic with phospholipids. A part of them play nicely with water, and a part of them doesn't like water at all, like traditional lipids. And so this combination makes for a very interesting functional property that lends itself to forming the cell membrane. Okay, we'll talk about that here on the next slide. So because of this unique structure of a part of it liking water and a part of it not liking water, when we have these two phospholipids coming together where their tails face each other, and their heads face the aqueous environment or the water that they're surrounded by. This creates this nice structural component that we refer to as the phospholipid bilayer. This is what lines all of our cells and helps to separate those body compartments that we talked about earlier in that first unit. So this is why we have an intracellular environment versus an extracellular environment, because of the separation that's created by these unique amphipathic molecules. We also see some other different structural molecules or structures that are created by phospholipids, such as micelles. Micelles are these kind of lipid droplets. They help to move fats around in our body, right? Remember, our bodies are made mainly of water, but we also need fats. Fats are quite important to cell structure, to creating hormones, which we'll talk about, to digestion, to energy. It's one of the most energy abundant macromolecules. And so when we think about the fact that fats have to move around in our bodies that are made of so much water, we use some of these structural molecules like micelles to help wrap those fats inside and help them travel around the aqueous environment in the body. Surfactant is another example of a phospholipid. Surfactant helps to keep our lungs inflated or help them preventing them from collapsing. Surfactant becomes really important for newborns because our lungs don't mature until the later parts of gestation, so 27, 28 weeks, 26 weeks even. If we have a premature fetus or a premature infant, then we run the risk of having undeveloped lungs or underdeveloped lungs. And so we typically give these neonates surfactant to help their lungs mature. And what surfactant does is it helps to cut down the surface tension in the lungs by, again, having this molecule that interacts with water on one side and then doesn't like water on the other side. And so it helps to keep the lungs inflated or help prevent them from collapsing. Okay, so some really important kind of everyday uses and functional important properties that we see with phospholipids. All right, so let me pause here and see if there are any questions. Okay. All right, so I don't see any questions. I'm going to keep going, but if you've got questions, definitely drop them in the chat box so I don't kind of run ahead of myself here. Okay, the next category of lipids are called eicosanoids. And eicosanoids are basically modified fatty acids. So when some of the fatty acids come in contact with particular enzymes, the enzymes cause them to kind of fold upon themselves and create these complex ring carbohydrate structures. So they're basically lots of carbohydrates that have folded themselves into these complex molecules that we call eicosanoids. Now, the function of eicosanoids is in intracellular signaling or cell communication. Examples of eicosanoids are prostaglandins, which are involved in our immune response. So these help to send signals from one cell to the other or tag certain cells for an immune reaction. As well as thromboxanes, and thromboxanes, we'll talk about them a little bit later down, but these molecules are involved in blood clotting. So again, they're going to be helping to send signals that help to form blood clots and help to repair injured vessels and so on. So basically molecules that can tag certain cells and help cells communicate with other cells, or a process we call intracellular communication. All right, and then the final category of lipids are steroids. I'm sure we've heard of steroids in some way or other. Steroids are mainly derived from cholesterol. Cholesterol is a substance, a waxy fat substance that our bodies make, our bodies need. All of our cells have cholesterol. It's important for digestion. It's also a main precursor for many important hormones in our body, things like estrogen, testosterone, aldosterone, vitamin D is mainly made of cholesterol, thyroid hormone. A lot of our hormones are made mainly of cholesterol, which is one of the main steroids. In terms of the structure of a steroid, it's basically ring. It's a complicated ring structure, and that's what offers some of the rigidity and flexibility to the cell membrane as well. And we'll talk about that when we look at the cell structure. Next.